Hi, everyone, and welcome to today's MPG Primer. Today, we have a fantastic talk by Rand Sui on an introduction to fine mapping, which, as a human geneticist, I think this is going to be a great introduction on this really important analysis technique. So just to give you a little bit of background about Rand, she is a computational biologist at Broad, and she is also a member of the Analytic and Translational Genetics Unit, or ATGU, at MGH. Prior to joining the ATGU and Broad 7 years ago, Rand's PhD work was focused on mathematics. At the Broad, she worked with Dr. Hilary Panuke to develop new fine mapping methods, and she is currently working on another fine mapping project that we might hear about today. Thank you.
Thank you for the introduction. Thanks for coming to this MPG Primer. So introduction to fine mapping. This is a very, this is an entire field of research that goes back at least a decade. And I'm not going to cover most of the important topics, and I'm not going to focus on the specific methods really, but today I decided to focus on what I think are some basics about fine mapping. So my goal today is for you to leave with somewhat useful framework and enough confidence to go on your next interaction with fine mapping, whether or not that is to apply methods or to read some papers or to do method development, or you just have some fine mapping results that you want to interpret. So that's my goal, and you might expect a somewhat slow pace in the beginning, at least. But I would like, there's some kind of echo. My goal is for you to be able to follow. So feel free to stop me with questions whenever you have them. Okay, so let's get started on telling you about the overview of this talk. So we can't really talk about fine mapping without talking about GWAS. And I think maybe most of you have heard of GWAS or interacted with GWAS before. So I'm just going to review GWAS and just go one level deeper into the basic math of GWAS so that it's a little bit easier for me to explain to you what GWAS can or cannot do. And then that leads directly into the goal of fine mapping, which is basically to do what GWAS cannot do. And then I will talk about how do the current algorithms typically quantify this goal. And in that section, I want to answer this question that I had 5 years ago when I was first introduced to fine mapping: is why are most fine mapping methods Bayesian? And hopefully I have a good answer to that right now. And I want to introduce you to the definition of two pretty important outputs of fine mapping algorithms. One is posterior inclusion probability, often called PIP, and credible sets. And after that, I want to talk about how do algorithms achieve this goal. And first, we'll start with a simple case of single causal variant method, ABF. And then talk about the pros and the cons, and that leads directly into the motivation for modeling multiple causal variant. And then after that, I want to talk about this topic that I don't think is talked about often enough, which is how to evaluate fine mapping methods. And last but not least, I want to talk about multi-ancestry fine mapping, and there's basically 3 approaches to do that. So, okay, let's get started on GWAS. So this is a Manhattan plot that I basically pulled from Wikipedia, which you probably have seen before, and it's very simple and elegant. The taller the point is, the more associated a variant is to your target trait, and it gives you a direct view of which loci you should prioritize when you're studying the genetic underpinning of disease or traits. So just to go a level deeper into the math, I want to just briefly go through how you obtain this Manhattan plot, basically how you generate these minus log 10 p-values for each variant. And so the way of doing that is basically you fit a simple linear regression. So simple means that you only have one feature. So this equation here, y equals to xj times beta j plus e. Your y is your target vector and whichever phenotype you prefer. And your xj is your genotype vector at one position, SNP-j. So you can imagine this could be the number of copies of a T allele at a position. And it's coded in 0, 1, and 2. So you plot this, you gather the data and you plot this scatter plot. And the process of fitting a simple linear regression is basically to draw a best fitted line through this set of dots. And the math is very simple as well. So the estimated beta j, basically the estimated slope of this line, is very easily expressed with a simple vector product. So this beta j hat is simple to compute, and oftentimes you want to know the standard error of that estimation, and that is sj, and which is also very easy to compute with a simple vector multiplication. And if you further want to know how significant this association is, basically how deviated this beta j hat is from 0, because the more deviated it is, the more associated a variant is to the phenotype, you would want a p-value. So before you compute the p-value, you want a z-score. So z-score is basically you divide the beta j hat by the standard error to produce a z-score, and then from there you can compute a p-value, and the smaller this p-value is, the more associated the variant is to the phenotype. And for visualization purposes, you flip that around to take a negative log 10 of the p-value, and that means the bigger this number is, the more associated the variant is to the phenotype.
Rand?
Yeah.
Rand, there was one question in the Q&A asking why regression was used for GWAS instead of chi-squared to generate these p-values.
Oh yeah, chi-squares are often used as well. So I believe chi-squared is just the z-score squared under certain assumptions. So yeah, you can use that as well. I think often when I'm computing p-value, I use the chi-squared to do it.
Thank you so much.
Yeah. So you may already know this, or you may be able to deduct from the math that GWAS results merely measure associations. You cannot use it to do causal inference. So how do we fix this? And what's the problem? So here I have a very simple example to illustrate the problem and to give you a solution, maybe. So here is a very simple data set with only 7 data points. And this is the data set that you observe with 2 features, x1 and x2, and a target. And the unobserved ground truth, which you don't know, is that x1 is causal, x2 is not causal. So you can see, actually, this data set is simple enough. You can see that x1 is perfectly corresponding with the target. But okay, so for an easier intuition, maybe I like to think about realistic situations. Suppose that x1 is the number of dishes that you ate for lunch, and target is the fullness index that you measure after lunch. 0 is you're very hungry, and 1 is that you're full. So clearly there is a causal relationship, right? So you ate a dish, you're full. You didn't eat, you're hungry. But x2 is interesting. x2, you can see that it corresponds to the target a fair number of times, but it's not perfect. And I told you that it's not causal. So you can imagine x2 could be the number of times you steps into a restaurant at lunchtime, right? So it is not causal to how full you are, because you can't expect to step into a restaurant, step right out to become full. It's not causal, but it is correlated with the number of dishes you eat for lunchtime, because you often eat your lunch at a restaurant. All right, so if you do a simple linear regression with x1, you just gave the model x1 to look at, your beta 1 hat is going to be 1, and the significance is going to be very significant. But if you do a simple regression with just x2, without giving the model to look at x1, with the target, the model is going to try to fit this line to its best knowledge, right? It's only looking at x2. So you're going to get beta 2 hat to be pretty close 1 to 1 as well. And the p-value is going to be pretty significant. And this is purely because x1 and x2 are correlated. And you cannot use this to do causal inference. So what can work is that you can use multiple regression to fix this. You can let the model see both x1 and x2. And so upon giving the model both vectors, it immediately figures out that it does not need x2 at all. So x1 beta 1 hat, the joint beta 1 hat is 1, and the joint beta 2 hat is 0, basically. I didn't make these numbers up, by the way. I did run this linear regression in Python. And if you now compute the joint p-value, the x1 is still very significant. It's too significant for my computer to display exactly what it is. X2 has lost all its significance. So now you can conclude that x1 is causal, x2 is not causal. So that's basically the not exactly how fine mapping is done, but it's the philosophy behind fine mapping. And so the goal of fine mapping is to find the causal variance, to pinpoint the causal variance. And you have this observed GWAS. It is a version of the underlying ground truth, but it's an obscured picture of underlying ground truth. It's obscured by LD, by noise, and by other factors. So the goal of fine mapping is basically to reverse that and to find the ground truth. So at this point, all this talk about multiple regression versus simple regression, if you were me 5 years ago, you might wonder, okay, why don't we just do a multiple regression then? Like if simple regression doesn't work, let's just do multiple. And I did ask my PI this question 5 years ago. I asked multiple times, and she was very patient with me. And she explained to me that, you have oftentimes you don't have enough individuals for your big locus, like you have tens of thousands of SNPs and your sample size is less than that. And if you have a wide data set like that, then you wouldn't be able to do multiple regression, basically. And then I kept asking because I don't want to accept this answer. I already know how to do multiple regression. Why do I have to learn about Bayesian methods? And I got another answer. So, okay, so even if you got enough individuals, sometimes there's this weird structure in the genome that a lot of the variants could be very, very correlated. And that could destabilize your multiple regression. And it just wouldn't work that well. She didn't say it totally wouldn't work. And later I found out there's actually a lot of frequentist method to do fine mapping. But the majority of the researchers, I think, has settled in on Bayesian for a lot of good reasons. And I kind of want to just bring out this example to sort of motivate the Bayesian fine mapping method. Okay, so I simulated a region with only 5 SNPs. And the truth is that all these 4 SNPs on the sides are not causal. They have 0 true effect sizes. And only this middle one has a non-zero true effect size, 0.15. So I use this true effect sizes and the genotype that I generated and this equation to simulate the simulated phenotype. And one interesting fact about this region is that I set it up so that all the variants are very correlated. In fact, the pairwise LD is 0.99. That is a pretty tricky situation. So you can imagine, even though the truth is that there's only one causal variant in here, if you do a simple regression with all the variants, individually, separately, you're gonna get pretty significant minus log 10 p-values for your GWAS, right? Because it's all correlated, the significance all come from this correlation to the causal variant. And in fact, when I did the GWAS, the minus log 10 p-value are all between like 42 and 50, which is way past genome-wide significance. So now, okay, I think it's a good time for multiple regression to step up. What happens if we do a multiple regression, right? Okay, in fact, we do have some estimated effect sizes, but the multiple regression is so unstable that it has very big error bars, because the algorithm can sometimes overestimate one effect size for one variant, and then it'll compensate by underestimate another effect size for another variant, because they're basically interchangeable. The correlation is 0.99. So this results in pretty wide variation in the estimation of effect size. And as a consequence, you can imagine that if you're trying to compute z-score on your p-value, what happens is that this is the joint p-value, and it's very not significant. You can't get anything out of this data, right? This yellow line is the 0.05 significance threshold. None of it passed the threshold. So now do you say that, oh, maybe there's just nothing here. We don't want to prioritize any variant in this space. But in fact, I want to show you like what Bayesian fine mapping can do. I'm getting ahead of myself a little bit because I haven't defined either ABF or PIP, but just know that this y-axis represents a probability of causality. The higher it is, the more we think the variant is causal. So what the Bayesian method can do is just to put a prior on this locus, because there's obviously something here. The GWAS is very significant. So maybe we'll put a prior on maybe just one effect. There's only one causal variant here. Let's not go crazy. While you're at it, don't estimate the effect size to be too big. So this has a stabilizing effect to the algorithm. So that's what Bayesian methods can do. And then this probability of causality right here is telling you another piece of information also. Is like, this locus is too complicated. I'm not super sure about anything. The probability of causality is hovering around 0.5, and I'm not giving it too high of a confidence. So it has a very intuitive way to kind of quantify your understanding of the posterior probability for causality, right? So there are some advantages to Bayesian fine mapping, like because it can incorporate the priors, but the ability of incorporating priors is kind of both a blessing and a curse. And the curse part is that all priors are basically intrinsically subjective. Your posterior probability is going to be not great if your prior is not great, right? So, but on the bright side is that a good prior can be very helpful. And if I'm not convincing you here, I'm going to convince you with this XKCD comic. So two statisticians, one frequentist, one Bayesian, and they're looking at this device which can detect whether or not the sun had exploded. Okay, and what this device does is that it rolls two dice, and if they both come up 6, the device actually lies to you, but otherwise it tells you the truth. So it rolls and it comes up yes. Okay, so now the frequentist statistician start to compute p-values. Okay, so the probability of the result happening by chance is 1/36, right? To roll 6 on 2 dice. And it's 0.027, and it's lower than the p-value cutoff, 0.05. So I conclude that the sun had exploded. But what the Bayesian can do is to incorporate prior. So what is the prior? What is the prior of the sun exploding? It's basically 0, right? So you can incorporate that prior with your observed data and then produce the updated probability for your belief of the sun exploding, which is for the Bayesian still pretty close to 0, because in this case, the prior dominates. Okay, so hopefully that is convincing to you that Bayesian fine mapping is the way to go. With that in mind, now let's talk about the Bayesian fine mapping outputs. So with the goal of recovering this causal structure, Bayesian fine mapping method had 2 important outputs. One is the PIP, which is depicted on this y-axis here. The higher it is, the more causal we think it is. And the definition of PIP is the probability that a SNP is causal given the observed data. And another very important output is the credible set. So the credible sets, the idea is try to capture the uncertainty of the identity of the causal SNP due to LD. So you can see that this toy example had outputted 2 credible sets, which you can think of is to correspond to these 2 causal variants. And it had basically recovered the ground truth, but there's certainly some complexities that it's taking into account. For example, this credible set 2, between all these SNPs, there's very tight LD. This credible set is basically the algorithm is trying to tell you that there is one causal variant in this set, we believe, but we're not sure which one it is because of how closely correlated they are. So that's the idea for credible set. I would say that there is another way to define credible set, which is I think actually the more common way among the available Bayesian fine mapping methods is to try to capture one credible set per locus, and this credible set will try to capture all the plausible causal variants with certain confidence level. And I feel like even though that's a more common way to define it, this new way of defining credible set, I think invented by the Suzy method, is the way that I prefer to think about credible sets. So this is what I'm presenting you. But if you are trying to interpret some fine mapping result, just be careful which method that they are using and what's the definition of credible set for that method. Okay, so now we're ready to talk about Bayesian fine mapping methods. I am definitely not trying to be, comprehensive here. This is not even close to half of the available methods in the field, but I am just picking out a few of the maybe more well-known. Also, the definition of well-known is like my subjective view. So I'm picking those out to introduce to you, and I've grouped them in 3 categories, basically, based on how they think about causal variants. So the first category is this approximate Bayes factor method, which only assumes single causal variant per locus. This is a very widely applied fine mapping method, perhaps the earliest fine mapping method. It's got a lot of advantages. It's simple, easy to apply. It only needs GWAS data. It does not need the individual level data. But there is some, with that simplicity, there comes surprise. And I will talk about that a little later. So first, I want to point out this the little trivia fact that the word approximate is actually optional. And it's only mandatory if you're dealing with case control traits. For quantitative trait, the Bayes factors are exact. Okay, so what are Bayes factors? So Bayes factors, you may have heard of it. If not, it's pretty intuitive to understand, actually. It's a likelihood ratio. It's basically saying that how much more likely is the data to be observed given that SNP I is causal than given no SNP is causal. So this Bayes factor can be, okay, maybe 1-fold, 10-fold more likely given this causal model over non-causal model, or maybe it's 100-fold more likely. And your Bayes factor for SNP I would be either 10 or 100, or it could be thousands. And this is quantity that this method uses because it's actually pretty easy to compute. You can implement it today with Python, like 4 lines of code, basically. And this Bayes factor also has an important property that through a pretty simple derivation, you can translate this into a posterior inclusion probability for each SNP in your locus. And just by normalizing all the Bayes factors so that they add up to 1, reflecting the fact that there is only one causal SNP in the locus. Okay, so some properties about Bayes factor and PIPs obtained this way. So first property is that the Bayes factors are roughly monotonic transformations of z-scores. That means that it does not change the ordering of the z-scores. The highest z-score, you will get the highest Bayes factor. If your SNP has the lowest z-score, your SNP will get the lowest Bayes factor. And I simulated, simulated a range of effect sizes in this locus so that you can see a range of d-scores over here. And you can see if I just compute the base factor using a formula that's pretty simple and then change it to log scale, this is monotonic. And also the transformation to PIP is monotonic as well. And an important consequence of this monotonic transformation is that the highest PIP is almost always given to the most significant variant. So you take a locus and you look at the Manhattan plot, and the top variant will pretty much always get the highest PIP. And one consequence of this is that it's very easy to construct a failure state for ABF. So that's what Qingbo and Hailiang did in their paper 2022. So they constructed this example where you have two causal variants violating the model for ABF. You have two causal variants, and there's a non-causal variant, a third one, non-causal variant. And the two causal variants don't really have any LD with each other, but they both have a non-trivial LD with this middle non-causal variant, LD being 0.3. So when you're doing GWAS, when you're just showing the model, this middle variant, and you're doing simple regression, it actually come up the most significant among the three. Because there is actually, if you're just doing simple regression, you're not showing the two causal variant, there is actually like an aggregating effect for the effect size, for the causal effect sizes in the middle here. And that is why it came up the most significant in this locus. All right, so what happens to ABF now, maybe you can take a guess, is that if you apply ABF on this 3-point locus, ABF will assign the top PIP to the non-causal variant, right? And therefore producing a false positive. So this is the motivation for modeling multiple causal variant, because we do have evidence suggesting that single causal variant per locus is not sufficient. It's usually not true. And if you just model single causal variant, you will come up with false positives like this. Okay, but the problem is that jointly modeling multiple causal variant is computationally challenging. So suppose you have 10 variant, just 10 in your locus. And if we assume a single causal variant, we only need to consider basically 10 possible causal configurations. These causal configurations are basically represented by these 0, 1 vectors. 1 indicate causal, 0 indicate not causal. So, and all of a sudden you want to consider 2 causal variant, right? So now we have to consider 45 possible causal configurations. And you have to compute the base factor for all of them in order to normalize your base factor into PIP. And you can imagine it goes out of hand pretty quickly from there. If you assume arbitrary number of causal variant for a locus with m SNPs, then you need to consider 2 to the m configurations. And that's the number of base factors you need to compute in order to normalize into PIP. Okay, but the good news is that there are algorithms, people had thought really hard about this, and they had come up with some solutions. So Caviar, I believe it's a big milestone in fine mapping when it's published. I believe it's the first method to jointly model multiple causal variant, as well as allowing summer statistics for input for fine mapping, and that is kind of a big deal. And the way that Caviar dealt with this computational challenge is by limiting the number of allowed causal SNPs in the locus, and also limiting the size of the locus. So you can imagine that reduce the number of configurations you have to consider. And there's improvements of this family of methods, Caviar BF, Caviar. So I remember fondly about the paper Caviar BF. It helped me a lot when I first started trying to understand the algorithms of fine mapping. I recommend you to go back and read that as well. It's a very well written paper. The derivations there is pretty clear. And then after that, we still need to model more causal variant, and just by limiting the number of causal variant per locus to, I think, 3 for Caviar is not quite enough for us. So there comes FineMap and DAPG. What the idea was basically to kind of restrict to a set of prioritized configurations to compute the base factors for in order to normalize the base factors into PIPs. So the way fine mapping does it is through a stochastic search algorithm. It's very efficient, and it can allow up to about 10 or even more, really. You just need to spend more time computing 10 causal variant per locus, and DAPG does a deterministic approach. So Susie kind of came along a couple years ago, and it uses, let me think about iterative Bayesian stepwise selection algorithm. It's a little bit different from the idea for these previous methods, and it's very efficient. It kind of utilizes the elegance of ABF and conditional analysis without losing the rigor of jointly modeling multiple causal variants. And the model is similar, but it's sufficiently different. I feel like it deserves its own bullet point. Anyway, so the last set of methods is actually came from our lab. We have realized that with increasing sample size and increasing evidence for basically polygenicity, you can't really define causal variant the way you define it for the previous methods. So the way previous methods define causal is variance with non-zero effect size toward the phenotype, right? So if you have polygenicity, especially high polygenicity for some of the complex traits, you can't really say like a lot of all these variants have zero effect, because they actually don't. A large proportion, or maybe not large, but a non-trivial proportion of the genome actually has non-zero effect sizes. And they may not be big effect sizes, they might be very small. And when you're trying to develop therapy for some disease, you may not want to think about them. But computationally, if you leave them unmodeled, it can actually mess up your discovery for those large effect size variants. It can affect the accuracy. So we found that that might be the case. So we came up with this new definition for causal. Is basically causal meaning that you have a larger effect among many small effects in the locus. So we changed the definition of causal, and then we implemented that model, and that is Susie Inf and FineMapInf, the new methods that we developed.
Ram, there was a question. First, the comment that before the question, just people are really enjoying your talk.
Oh, thank you.
Do you notice a bias for directly genotyped SNPs versus imputed SNPs when they've gone through the GWAS analysis pipeline?
Yeah. This is I think many researchers have thought about this problem. I have definitely worried about this, but there's just simply too many things to worry about, and this kind of fell off the priority list. But yeah, I believe there's definitely something there, and we should look more into the how does the imputation uncertainty affect the accuracy of our GWAS and therefore the downstream fine mapping.
Thank you so much.
Okay, so now I want to take some time to talk about evaluating fine mapping methods in first in simulations. So there are so how do you know if your fine mapping methods are good? Basically, there are two metrics that you can look at in simulations where you know the ground truth. One is calibration, the other is recall. One measures accuracy, the other measures power. So calibration is basically answering this question of variance with PIP X%, are X% truly causal? So here's an illustration of my understanding of calibration. So suppose you have a phenotype that you have fine mapped, and you have gathered all the high PIP variants, high PIP meaning PIP greater than or equal to 0.9, and you gathered all 10 of them. And so what the algorithm is telling you with this high PIP is that you should expect 90% of these variants are truly causal. This is what you should expect. So you then, look at the truths that you have in your simulation, and you discover that only 5 out of the 10 variants are truly causal. So your algorithm kind of lied to you a little bit. So this is what's called miscalibration. But on the other hand, if you look into the truths and your 90% of these SNPs that came up high PIP are truly causal, then your algorithm is calibrated, and that's good. So our group did some simulations with what we think are reasonably realistic simulation settings. And we tested a couple of fine mapping methods, SUSY, FineMap, ABF, CODRO plus ABF, which I didn't introduce, sorry. It's a conditional analysis based approach, allows for multiple causal variants in the locus. And we simulated like genome-wide structure that are realistic, and we kind of plotted this calibration plot, and the fact that all the colored dots lie on this sort of a diagonal line means that it's good news. It's calibrated. So what is recall? It's basically trying to understand what proportion of all the causal variants are captured by the X% variance with the highest PIP. So suppose you simulated some ground truths, and all you have that are causal are these 10 variants. And you run your fine mapping method on it, and you have this list of variants. You rank them from high to low based on PIP, and you take the top, say, 1% of the variants you have fine mapped. And you look at the truths, you find that you captured 4 out of the 10 causal variants that you simulated. So the recall here is 40%. And if you take the top 0.01% of variance is 20%, you capture 20% of all the causal variants. So therefore, with these numbers, you can kind of plot a recall line. And we did this for our simulation as well. And you can see here, recall is telling you a slightly different story, giving you a more full picture of the comparison for all these fine mapping methods, right? So here you can see this ABF and CODRO ABF has lower recall, almost just half the amount of the causal variants that are recovered by SUSY and FineMap. So this is saying that, okay, even though they are all pretty accurate, the calibration plot looked good for all of them. But here you can kind of see that the ABF method, CODRO ABF method, had lower recall, so they have lower power. So 2 metrics gives you a more complete picture of your fine mapping methods. But the problem with okay, so I only have 5 minutes left. Good, better. So the problem with simulation is that you really don't know how good your simulated conditions are and how much it reflects your real data, right? So there's a metric that we have developed in real data, which I unfortunately, I think I have to skip. It's easier to skip my own work than to skip other people's work. So basically, we thought that if we can't compute accuracy of fine mapping, then we can compute consistency. So we basically did 2 rounds of fine mapping for the same cohort with 2 different sample sizes, and we compared the PIP, and we found an alarming amount of inconsistency. And we also found that if we do this analysis and compute this inconsistency in simulations, it is not as alarming. It is not alarming. So that what that contrast is telling us, so what this bar plot is trying to tell you without defining anything on the bar plot, is that it's our simulation is not realistic enough to reflect the complexity in the real data. So this is kind of good to know. It gives you motivation to develop more, maybe realistic models in order to get better fine mapping results. So that's all I'm going to say about that. And then we'll switch gear a little bit and talk about multi-ancestry fine mapping. Okay, so why are we interested in multi-ancestry fine mapping? There are many, many reasons, but from a computational scientist perspective, from my perspective, is that the LD patterns basically, and the increased sample size, of course. So from the beginning of this talk, you might remember that more LD gave us more trouble, right? So the flip side of that is that less LD gave us less trouble. So in diverse ancestries, sometimes, oftentimes given the locus, if you plot the LD for this locus in different ancestries, you will find that they have different patterns. And then you might be really happy to find that in some population, the LD seems really minimum. And that is very good because now you can really distinguish each variant, and you can really try to pinpoint your causal variants. So, and then, plus you have more samples, so more power to detect causal variants. So if that's what you want to do, if multi-ancestry file mapping is what you're interested in, currently you basically have 3 approaches. The first approach is the meta-analysis based approach. The way to do this is pretty easy to understand. You take your multiple GWASs from your multiple ancestries, and you perform meta-analysis to combine all of the GWAS into one GWAS. And then you take the average LD of all these ancestries you're analyzing and then produce average LD. And basically you use this combined GWAS and combined LD to perform single population file mapping. The pro is that it is very simple to apply. There's actually a lot of high impact paper that apply this meta-analysis approach to find causal variant, very effective. It improves discovery, although the downside is it does not assume any heterogeneity between populations. It basically just takes the average and use that. And the second approach is the post hoc analysis approach. So this one, you fine map each population separately, so you don't have to worry about how to model different LD structure, how to model all these complex things. You just do single population fine mapping. And then after you've done that for all the population you care to do, you combine the results using algorithms or manually. So the pros is that it gives researchers a lot of freedom to interpret the results, and it allows for population-specific inference. But the cons, one of the cons is that it doesn't take advantage of your increased sample size. You're still just doing single population file mapping, and there could be, power loss there. So there comes Bayesian statistical methods. This way, it models multiple cohorts directly, and it will fine map all the data all together. So the pros, you can easily imagine, it's a very principled way, rigorous way to do multi-ancestry file mapping. The cons is that it can be pretty computationally intensive, and it depends pretty highly on the accuracy of your model, how you model the relationship between effect sizes of different populations and so on. Okay, so so aside from all these computational challenges, we still have a lot of data recruitment challenges for multi-ancestry file mapping. So especially data recruitment on European ancestry groups is challenging. But I think there are a lot of really smart people, devoted people working on this challenge right now. And data harmonization could be challenging as well. Different genotyping protocols and different ways to do the phenotyping harmonization is challenging as well. And of course, the computational challenge is kind of what I think about most, is jointly modeling multiple ancestry can be very challenging. And I really encourage more researchers to go into this field of study, because this is part of the future of file mapping, is to do multi-ancestry file mapping. And with that, I conclude this talk. I want to thank Hillary for introducing me to file mapping and introducing me to statistical genetics in general. And I want to thank Zoe and Zach for their statistical expertise and endured a lot of my tedious questions through the years. And I want to thank Masa for teaching me about multi-ancestry file mapping. And I want to thank Josephine and Vivian to sit through my practice talk for this talk and give me very helpful feedbacks. And I want to thank Ben and Mark for their advice and for their steady support for my work.
If you guys have any questions, you can come to the mics. I will also be looking on the Zoom chat. I first want to just say thank you so much for a wonderful, very nice introduction. Not too much math, it wasn't scary. What aside from the multi-ancestry challenges in the future, what other things is the fine mapping field going to have to grapple with in the next 5 years or so?
Oh man, am I the right person to ask? So from my very narrow point of view, there's a lot to do for functionally informed file mapping as well, which I did not even mention. But that is the entire field of study on its own, just kind of producing useful priors to put in your Bayesian file mapping methods, like priors, kind of like, I believe coding variants are more likely to be causal than non-coding variants. And how do you, that's easy to say, but like, how do you quantify that? How much more causal? So, that is very important as well. What else? I wonder if there's, so I started thinking about something along the line of, PRS using fine mapping results, like polygenic risk score predictions using fine mapping results instead of using the GWAS effect sizes. Yeah, those are just to name a few. If the audience have more comments, I would love to hear.
And I was just wondering if from your perspective, do you have a ballpark of cohort size that you would make sure that people achieve either in their affected or unaffected comparisons before they think about applying fine mapping techniques?
Number of individuals. Yeah, so right now we have all these biobank size data. I think it's, I work with hundreds of thousands of individuals for any given phenotype, but there's actually like a lot you can do with tens of thousands. I'm not, yeah, I don't know exactly how I would quantify that. Yeah. Sorry.
Thank you for the great talk. I was really able to follow it after a very long time. So well done, well practiced. So you mentioned how in the recall, fan mapping and other tool or SUSY was better than COJO. What are the nuances we should be looking at? I mean, many times we don't have time to try each and every other tool to figure this out. So what should we do? So right now in our lab, we are trying COJO, but now I'm going to go back and tell them, oh, fine mapping is better than COJO, don't try COJO. But maybe for that particular study, COJO is better. How should we go about doing this? What should we look at? Maybe we need to try maybe two things, but what are the nuances? What should we look at from the GWAS point of view or some statistics point of view? And what we should run to get some idea or some hint which pathway to take? Maybe it's a broad question, but sorry, I have to ask.
Oh no, but that's a great question. I wish I know the answer. That would simplify my life a little bit. But yeah, so right now our recommendation is to run SUSY and FineMap, or SUSY inf and FineMap inf together, because they actually don't agree for some non-trivial amount of time. So what we want to do depends on what you want. Like if you want to be more conservative, if you want to be sure that the variants that you're following up are really the more causal variants, then you want to take the minimum PIP, like you want to go a more conservative approach, minimum PIP of all the methods that you run. And if you want to go a more like search discovery based approach, you can like take the, all the high PIP variants, well, include all the credible sets as well, if you want. So yeah, as to what method to choose, I really don't have a great answer. It could be study dependent, it could be phenotype dependent. If you have a highly polygenic phenotype, I recommend SUSY inf, FineMap inf. But if you are doing like EQTL studies, I don't think you need a highly polygenic structure. But maybe you're just, you're discovering things. Yeah, that's a really great question.
Sorry, one last thing. Multiple studies using multiple LDs, 5 kb, 1 kb, and now we are stuck. How to harmonize that? So what should be the approach here if you want to apply your idea? But guess what? We have different studies who have used different LDs. So we have different summary statistics. So how do we go about doing that? What should we look at now to help us to again make a decision when we have different pre-processing or post-processing, I don't know how to put it correctly, but before running these tools, things have been different and different LDs has been used. So how do we proceed now?
Yeah, I again wish I had an answer. But so one thing I would point out, maybe it's a little off topic for your question, is that we do recommend using like your in-sample LD instead of some reference panel, because that has been proven to be not great. You know, whatever data that you have, even if you can't get the individual level genotype data, phenotype data, you can do like an in-sample GWAS and an in-sample LD. If you really want the data to be clean, you really have to do all these QC by yourself and try to harmonize all that. But if you already have a couple of set of messy data to start with, I'm not sure.
Concludes the primer. Let's give Ran another round of applause.